Pangenomes are a new way to represent the diversity in a population or species, and they're useful because they manage to cluster together differences between individuals that were not possible to exploit before.
I started working on pangenomes about 2 years ago when I joined Tobias Marschall Lab in Düsseldorf. And I quickly got involved in the Human Pangenome Reference Consortium. And the work that is set out there is to construct a pangenome of up to 300 human cells.
Pangenome graphs are already useful for mapping reads, sequencing reads from other people, and they improve the accuracy of mapping, and they can also help us to find variation which was not found before. But I think we still have a lot of work to really understand the graphs themselves and to get information directly from the graphs. So I think this is something that still needs more work, and that's really interesting challenge for our future research.
So I've seen sort of 3 definitions of a pangenome. One which is older, which I think then you get into these technical terms like the peripheral genome, the core, everything like that. But I think what we mean is a representation of a population, the whole thing. So in particular, for example, if you look at the the pangenome, what is it? HPRC, Human Pangenome Reference Consortium. So they're actually saying, look, we really want to represent everybody. You get better results if you reflect the diversity of everybody, not just when you're studying humans, actually, when you're doing other species. But this is particular interest now because we have hundreds of thousands of human genomes, and we're not. I mean, we have them there. We're mining them, but we're not using them in all applications. For example, DNA alignment, because we're still figuring out how to do that, because there are technical issues about aligning against a representation of all of humanity's genetic diversity.
Well, I think the one thing you can agree on with regards to what a pangenome is that nobody quite agrees what a pangenome is. Pretty often, it seems that when people who work on data structures talk about a pangenome, they're talking about a data structure, as far as I can tell. But more applied people are kind of more so referring to kind of the collection of variation within the genomes of, say, a species, of a specific group. And I think that would be the definition that I'm most comfortable with and use the most. Like, for instance, in my work, I work on the level of the genes mostly, rather than, say, variation on the level of nucleotides. And so for me, a pangenome is core genes along with accessory genes.
Pangenomic field is really interesting because it puts together lots of people who are doing very different things, and all of them are sort of not satisfied with the way how we represent genomes and how we treat genomes, and they see a potential how to improve this. So it is really interesting to watch, and it will be interesting to see where this field goes in the next 5 to 10 years, and how we will move from looking at single genomes into a world where we are looking into genome of a population as a whole.